Hello, welcome to D-board exams biochemistry review. So we're going to review glycolysis and make sure you have gone through your dental decks, okay? And this is going to be a quick review of some of the concepts you're supposed to know before you go in this exam, okay? All right, so a summary, we have a summary talking about the ATP, okay, produced in the glycolysis, pyruvate, and of course you have to know some of the enzymes that characterize the main regulatory points of glycolysis. Okay, all right. So in glycolysis, a net of two ATPs are formed. Okay, now usually in glycolysis there are four ATPs which are formed, but you have to use two ATPs in order to produce ATP. So you are investing two ATPs, okay, and producing four ATPs. So that means you have gained two ATPs. That's what the net means, okay? It's net of two ATPs, all right? If they ask what's the gross number of ATPs produced, is going to be 4. Okay, but the net number of ATPs produced in glycolysis is 2. And then also we're going to discuss about how pyruvate has a different metabolic fate depending on the presence or absence of oxygen. Okay, so the cellular condition of the cell. Also, the regulatory point for glycolysis, okay, is characterized by the phosphofructokinase. Okay, so you have to know that phosphofructokinase is the main regulatory point of glycolysis. And then the hexokinase and pyruvate kinase reactions are also regulatory points, but not as important as the reaction which is characterized by phosphofructokinase. All right. So the fructose 2,6 bisphosphate is an important regulatory molecule, and we're going to see that. All right, so glycolysis is the process of converting glucose into pyruvate. Make sure you know that, okay? The purpose of glycolysis is to convert glucose into pyruvate. Glucose is a 6 molecule and it's broken down into half. So if you break down 6 into half, you have 3 and 3, right? So pyruvate is a 3 carbon molecule. Okay, glucose is a 6 carbon molecule, pyruvate is a 3 carbon molecule. So you produce 2 molecules of pyruvate. Okay, and there is a small amount of ATP which is produced. All right, so the net we said is 2 ATPs. All right, glycolysis. Okay, you have to understand glycolysis occurs under anaerobic condition. Glycolysis does not need oxygen. Okay, I repeat that glycolysis does not need oxygen. Glycolysis can occur in any cell without oxygen. Okay, the pyruvate. So also glycolysis, just remember the question that they like to say, the red blood cells. Red blood cells, remember, red blood cells don't have mitochondria. Okay, so they don't have mitochondria. So the only way they can obtain energy, produce ATP, is glycolysis. Okay, that's the only way they can produce energy. And then the pyruvate which is produced enters the citric acid cycle only when there is oxygen. Okay, so in aerobic conditions, that's when the pyruvate, which is produced in the glycolysis, enters the Krebs cycle, which is a citric acid cycle. All right, so that's a quick summary. Now let's look at some of the glycolysis in detail. Okay, so And these are the main points that of things that you have to know. Okay, the basics that you're supposed to know about glycolysis and energy production. So glycolysis is the metabolic pathway which is found universally in biological systems. Okay, so for biological system to produce energy, the glucose has to undergo glycolysis. Okay, glycolysis, of course, as you understand, is the breakdown of glucose. It is the metabolic pathway which converts glucose into two molecules of pyruvate. Remember, initially I said glucose is a 6 carbon molecule. So if you divide 6 into 2, you get 3. Okay, so it forms two molecules of pyruvate. Make sure you keep that in mind. One molecule of glucose produces two molecules of pyruvate. As a result, okay, a small amount of ATP and NADH are produced. So not only is ATP produced, but also we have NADH. So when you go through this, don't just memorize ATP, ATP. Also remember, okay, you have to remember which reactions are producing ATP. You have to remember which reactions are producing NADH. All right. Most of the metabolic energy derived from glucose come from the entry of pyruvate into the citric acid cycle. So um, you know that glycolysis only produces a net of two ATPs, okay? But once the pyruvate goes into the Krebs cycle, then you have more ATPs, okay? Depending on the system, you can produce over 30, okay? Over 30 ATPs. All right. So these pathways occur in aerobic condition. So if you have oxygen, that's when you can go into Krebs cycle. Krebs cycle needs oxygen. Okay. Okay. Needs. All right. So you have to know that. All right. So let's look at so the glycolysis occurs in anaerobic condition. All right. And in anaerobic condition, in the absence of oxygen, you can only make pyruvate, okay? And then which is converted, if it's in the muscle, you're going to form lactate. Remember lactic acid, if you haven't run for some time, if you have not exercised for some time and then you go running the first time without warming up, guess what you get? You start getting muscle cramps. That muscle cramps, that's because of the lactic acid which is being produced. Okay. So, however, in yeast, okay, in yeast, without oxygen, the yeast, the pyruvate is going to be converted into ethanol. These are key important points that you're supposed to remember. Okay, in muscle, in the absence of oxygen, you form lactic acid, which is lactate. Okay, so lactate. In the absence of oxygen, in yeast, pyruvate is converted to ethanol. Now, you have to know that lactate and ethanol are both 3 carbon compounds. Okay, they both have 3 carbons. So you have to understand that this pyruvate, which is also a 3 carbon compound, is converted to other 3 carbon compounds. Okay, very important points here. The other important point is so the conversion of pyruvate in the absence of oxygen to ethanol, okay, is called fermentation. Make sure you understand what the definition of fermentation. The conversion of pyruvate, okay, into ethanol. All right, is called is called fermentation. Okay, and this fermentation process, okay, can occur in the absence of a living cell. So you don't need fermentation to occur within the cell. It can happen anywhere. That's how beer is made, right? I remember we used to make uh, wine, wine from bananas. Okay, you take bananas, you you smash them, you add some water, you add a little bit of just a little bit of sugar, not too much, and then from there you add yeast, okay, the same one you use for baking, and then you just let it ferment, okay, and from there you form a wine, banana wine, okay. So you can. This is because the process of fermentation does not need a living cell, okay. All right. So um, but sometimes you don't even need the yeast to have fermentation. You can just put it on the side and let it sit, and you can produce some wine. So fermentation, okay. I elaborated on this process so that you can memorize it, okay. So let's look at some of the definitions of the thing you're supposed to know, okay. An enzyme. So an enzyme is a heat labile, non-dialyzed. So meaning it's not broken down, okay. It's a non-dialyzable substance. A coenzyme, which is most of the time it's like vitamins or some of the minerals, is a heat stable, okay, dialyzable substance. Now, when you say heat labile, meaning when you add heat to that area or to the substance, it's going to change conformation, to change shape. Okay. Now remember that if you add heat to certain enzymes, they're going to 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 speed up the reaction. Okay. Enzymes, they catalyze they catalyze the reactions. They speed up reactions. So glycolysis, you have to know the other name of glycolysis is Emden-Meyerhof pathway. Okay, it's something you need to know, but it's not that critical in terms of the board, but it's something you need to know. All right, so some of the points you have to remember. Remember that the lactate and ethanol are products of anaerobic condition. Okay, in the absence of oxygen, you form ethanol and lactate. Okay. Um, if somebody is sick, okay, they have shortness of breath, or if somebody was in trauma, okay, or just not receiving enough oxygen. For example, they're in shock. If somebody is in shock, then because shock by definition, the energy is not producing oxygen because of inappropriate delivery of blood to that area, you're going to form more lactic acid because of the absence of oxygen. Okay, so you can measure the lactic acid to confirm what's happening. The Now, when you don't have two key points here, okay, when you don't have when you don't have oxygen, you form lactate and ethanol. When you have oxygen, you form carbon dioxide and water. Okay, these are the products of aerobic respiration, which is the reason you can breathe on your arms, or you can breathe on a glass, and you have some of the moisture coming out. So you are releasing carbon dioxide, and also that water vapor condenses on the glass, and you can see that. That's because the product of respiration, okay, the product of respiration is carbon dioxide and water. And respiration in this case is when you go through to the Krebs cycle, okay? Because to go through the Krebs cycle, you need oxygen, and oxygen is going to be the electron acceptor once you enter the electron transport chain, okay? All right. So let's look at the overview of glycolysis here. So as we mentioned, you have 6 carbon molecule. Please memorize this formula also. You see this is simple, right? C6H12O2. Okay. Sometimes they can say the simplest form of this molecule. So if you divide here 6, okay, you divide 6 here, you divide 6 here. So it's going to be CCH2O. Okay, that's the simplest. All right, the simplest monomer or molecule that you can break down glucose. But what I'm trying to say here is you're supposed to memorize this formula C6H12O6. Very simple to memorize. Okay, so this is glucose, and then it's going to be broken down into pyruvate. Okay, now this is one molecule of glucose, but the pyruvate which is produced, because it's 3 carbons, okay, because it's 3 carbons, it's going to it's going to be 2 molecules, okay, 2 molecules of pyruvate. So here you can form this pyruvate can be converted to lactate. Here it shows 1 lactate, but remember it's 2 because you're converting 2 molecules of lactate. Pyruvate into lactate. Now this is when you don't have oxygen, so there's no oxygen in this here. Okay. Again, here no oxygen, so you form ethanol. This is in maso. This is in yeast. If you have oxygen, this area here, then you form carbon dioxide and water. Okay, so this is a quick summary of what we just discussed. Make sure you look at it. It's very simple. Don't bother yourself looking at the configurations here. Okay, just make sure you know that, okay, this formula of glucose is broken down into 3 carbon molecules of pyruvate. And if you don't have carbon dioxide, if you're in maso, you're going to form, if you don't have oxygen in maso, you're going to form lactate, and in yeast, you're going to form ethanol. If you have oxygen, then you're going to form carbon dioxide and water. Okay. All right. So here's a question. One of the products of the metabolism of glycolysis under anaerobic. Okay. Make sure when you read this, make sure you know what you're reading. Don't just go through quickly and think maybe they're asking aerobic. They're asking anaerobic condition is lactate. So true or false? That is true, right? We just talked about that. If you don't have oxygen, then pyruvate is going to be converted to lactate. So the major function of glycolytic pathway is to do what? Why do we need glycolysis? Right? Glycolysis is essentially, so is this to produce carbon dioxide? No. Okay. Because remember, glycolytic pathway does not need oxygen. So carbon dioxide is only produced if you have oxygen. Okay. And so does it directly produce most of the ATP needed for the cell? No, this is false, because glycolysis only produces the net of 2 ATPs. Okay. Does glycolysis produce glucose? No, this is false, because glycolysis actually breaks down glucose. So now we come to D, right? So glycolysis converts glucose to pyruvate. This is true. That is the correct answer. Okay. So don't be confused. If you didn't know, you can always eliminate what doesn't make sense, but make sure we already reviewed. Glycolysis breaks down glucose to form pyruvate. Okay. All right. So glycolytic pathway converts 6 carbon molecule into two interchangeable 3-carbon intermediates. We've already discussed that. Okay, I don't mean to make a line through that. So these 3-carbon intermediates are then converted to pyruvate. Okay, and all the structures which are involved in glycolysis contain either a 6-carbon ring, okay, or a 3-carbon ring. Okay. Now, many of the structures and reactions which occur as part of the glycolytic pathway, they are also found in other forms, okay, in other metabolic pathways. So it is very important that you understand these structures and reactions, okay, before you go any further. Now, for the purpose of this exam, you have to understand, okay, a 6-carbon glucose is broken down to 3-carbon molecules, okay? You have to understand that. That's the only concept you need to understand. You don't need to memorize all the sticky figures and confuse yourself, okay? Just the basics now, all right? All right. So, 6-carbon molecules are derivatives of glucose and fructose. Remember, glucose is 6-carbon, and also fructose is a 6-carbon. Okay. Now, the difference between fructose and glucose is that fructose actually is a 5-membered ring. It has 6 carbons. Now, you have to understand, fructose and glucose both have 6 carbons. Okay. Glucose is a 6-membered ring, but fructose is a 5-membered ring. It has 6 carbons, but the ring has 5. It's a 5-membered ring. Okay? You have to remember that. So, what are the derivatives of 6-carbon molecules? Okay? So the derivatives of 3-carbon molecules. The 3-carbon molecules, the compounds which are 3-carbon molecules are involved in the glycolysis are the hydroxyacetone, okay? Glyceraldehyde, very important to understand, okay? So these two and these two, very important. Of course, pyruvate is supposed to know really well, right? So all these structures listed here are 3-carbon molecules, okay? So make sure you know which ones are 3-carbon molecules, because okay, the boards like to ask, okay, which of the following is a 3-carbon molecule, and they give a bunch of molecules, okay? And so, in related to glycolysis, related to glycolysis, then you have the hydroxyacetone, glyceraldehyde, glycerate, and pyruvate. Okay. So let's talk about the enzyme definitions. You're supposed to know if you're going to go through the glycolysis. You have to understand, okay, the definition of enzyme. What does an enzyme do? All right. So a kinase is an enzyme which is responsible to transfer a phosphate, okay, a phosphate from ATP, okay. So a kinase is an enzyme which is responsible for phosphorylation, okay. If you are going to add phosphate to a molecule, then you use a kinase, okay. Kinase, phosphorylation. Kinase, you're adding phosphate. A mutase is an enzyme which is responsible to shift the phosphate group, okay, from oxygen within the same molecule. All right. Make sure you understand these definitions. They make it very easy for you when they give you a question. Isomerase. So isomerase, meaning you are going to make this a different configuration of the same molecule, right? So isomerase is responsible to convert a ketose or a ketone to an aldose, okay, to aldehyde, or vice versa. It can also convert an aldose into a ketone. So you have to understand that. A dehydrogenase. What does a dehydrogenase do? Okay. Dehydrogenase is responsible for elimination of water or dehydration. Dehydrogenase, dehydration, remove water. Okay. Aldose is responsible for splitting carbon bond. Okay. Aldo cleavage. So make sure you understand those. So let's do a question here. A quick review question. The enzyme which catalyzes a transfer of a phosphoryl group from ATP to another compound is called what? Is it a mutase? Is it a kinase, aldolase, or hydrogenase? Right? So this, okay. Actually, I'm a biochemistry major. That's what I studied in. I concentrated on in my undergrad when I was doing that. So I love biochemistry, right? So here we just discussed about the enzyme definition. So you have to know which enzyme transfers a phosphate, okay, from ATP onto a molecule, all right? Or is what enzyme is responsible for phosphorylation? So it is kinase, right? So make sure if you choose kinase or your kinase was in your mind, that's the correct answer, all right? If you chose mutase just because it shows green, then sorry, okay? All right, but so make sure you understand these enzyme definitions, okay? So quick reviews here. So, which of the following is not a 3-carbon compound, which is part of the glycolytic pathway, right? So, all of this, which is not a which is not a 3-carbon compound. We just reviewed which ones were 3-carbon compounds. So this is a 3-carbon compound, 3-carbon compound, 3-carbon compound. So you know fructose, okay, has a 6-carbon compound, but it is a 5-membered ring, okay. So fructose is not a 3-carbon compound. All right. So some of the steps of glycolysis that we're supposed to know. All right. So glycolysis takes place inside cytoplasm. Okay. Here I paused before I say cytoplasm so you can remember. Okay. This is one of the favorite questions that okay, where does glycolysis occur? If you have read through the dentodex, okay, then the thing that you should carry out, sometimes when you read the dentodex you may forget things, but the main purpose here, you have to know glycolysis occurs inside the cytoplasm. And then the electron transport chain or oxidative phosphorylation occurs inside mitochondrion, okay. So you have to know glycolysis occurs inside cytoplasm. The goal of the initial reaction of glycolysis is to convert, okay, glucose into fructose 1,6 phosphate. So this conversion traps glucose into the cell as glucose 6 phosphate and forms a phosphorylated compound, okay, which is fructose 1,6 biphosphate that can be cleaved. So when you form the glucose 1,6 bisphosphate, you can cleave it to form phosphorylated 3-carbon intermediates, okay, which is dihydroxyacetone and glyceraldehyde, okay. So these 3-carbon compounds, that then broken down, okay, to make ATP by the citric acid cycle and oxidative phosphorylation. These are the key steps, okay, and we're going to go through that quickly, okay. So this is what happens. Okay, so we have a hexokinase, right? So kinase does what? Kinase adds a phosphate, right? It adds phosphate. So look at that. You take glucose and ATP, okay? Now, this here, so 1, 2, 3, 4, 5, 6, okay? This carbon here is carbon number 6 when you start counting from here, all right? That's carbon number 6. So this hexokinase is going to take one of us phosphate. Remember, ATP has 3 phosphates, right? That's why it's called adenosine tri as in 3, triphosphate. So it's going to take one of the phosphates here and put it here. So it's going to be called glucose 6 phosphate because this phosphate is on carbon number 6. Okay, so you see you took a phosphate here and you put it here on carbon number 6, so it's called glucose 6 phosphate. Now, and then of course, then you form ADP. ADP is adenosine diphosphate, right? So now since you took off one molecule of phosphate, now you are remaining with 2 molecules of phosphate on, so it is ADP, right? Now, this glucose 6 phosphate, it cannot leave the cell, okay? So this traps glucose inside the cell, so it doesn't keep being transported out. Now remember, there are so many ways that the glucose can be transported. Facilitated diffusion is one of them, right? So you don't want uh, the glucose to be transported inside the cell and then come out, okay? Because you're not going to produce enough energy because it keeps coming out. So you have to trap it inside the cell. To trap it is you form glucose 6 phosphate, okay? So quickly, the first step in glycolysis, make sure you understand the first step. The first step in glycolysis is the phosphorylation of glucose by ATP to form glucose 6 phosphate. Favorite question, okay? The first step is catalyzed by hexokinase. The first step of glycolysis is catalyzed by hexokinase. If somebody wakes you up in your sleep real quick, what's the first step? The first step of glycolysis catalyzed by what? Hexokinase, right? What does it form? Glucose 6 phosphate or G6P, right? So quickly, these are the basics of you need to understand before you go in this exam, okay? You don't need to understand all the intricacies of biochemistry or all those pathways and synthesis. Just make sure you know the basics. All right. The second step in glycolysis is the isomerization of glucose 6 phosphate to fructose 6 phosphate. Okay. And this usually converts the sugar from a 6-membered pyranose into 5-membered furanose. Remember, I told you glucose is a 6-membered ring and fructose is a 5-membered ring, but they both have 6 carbons. All right. This reaction is characterized by phosphoglucoisomerase. Okay, so it is still the same molecule which has six carbons, but now the presence of the positions of the phosphate or the OH group is different. Okay, so you have to know that. So this is the reaction. So phosphoglucose isomerase, you get G6P, glucose 6 phosphate, you transform it into fructose 6 phosphate. Okay. they still both have one phosphate, but the position of the phosphate is different. Because now, even though it looks the same here that it's on this carbon, but it's different now. Okay? All right. Because now, so now you start counting from here. This carbon is number 1, right? Then 2, 3, 4, 5, 6. Okay. Now in a ring, make sure you know that this point, each point here is a carbon. If you haven't realized already, okay. And sometimes they just write the C for carbon, okay. So in this point, each point here is a carbon molecule, all right, which is the reason I am counting as 1, 2, 3, 4, 5, 6, okay. But you have a 5-member, they both have six carbons, but this one is a 5-membered ring, okay. But you still have the phosphate on the 6th carbon, all right. So phosphoglucose isomerase, all right. The third step in glycolysis is a second phosphorylation, okay? So now you have formed fructose 6 phosphate. Now you're going to add one more phosphate. Now to add one more phosphate, what enzyme is needed? It's a kinase. Remember, the kinase is the one which adds phosphate, right? So you need an enzyme. In this case, the enzyme is called phosphofructokinase. 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 It's a kinase, meaning it's going to add a phosphate onto a fructose molecule, right? That's where the name comes in. So if you look at the name, you understand, oh, okay, it's a kinase, so it's adding a phosphate. On which molecule? Okay, the molecule is given in the name, fructose, right? So you add phosphofructokinase, adds the molecule onto fructose 6 phosphate and forms fructose 1,6 bisphosphate. Okay, remember it's 1,6 because the phosphates, when they ask you where are the phosphates on fructose 1,6 bisphosphate, it means the phosphate are on the first carbon and the 6th carbon. So the first carbon is this one, 1, 2, 3, 4, 5, 6, and you can see this is the phosphate group. This is a phosphate group. This phosphate group is attached to carbon number 1, and this phosphate group is attached to carbon number 6. So it's called fructose 1,6 bisphosphate. Okay. So what enzyme adds a phosphate group onto fructose 6 phosphate? It is phosphofructokinase. Okay. 1-glucose is phosphorylated by hexokinase. Okay. Once glucose is phosphorylated by once glucose is phosphorylated by hexokinase, it can freely diffuse out of the cell. This is false, because remember, the purpose of the first step of glycolysis is to trap glucose inside the cell so that the glucose doesn't leave, all right, the cell. The first step of glycolysis is catalyzed by hexokinase by converting glucose into glucose 6 phosphate, and this traps the glucose inside the cell. All right? Hope you understand that. All right, let's look at another question. The conversion of fructose 6 phosphate to fructose 1,6 bisphosphate involves isomerization. Now, isomerization is just changing the phosphate group from one position to another position within the same molecule, right? But if you're adding another phosphate onto fructose 6-phosphate and you're forming fructose 1,6-bisphosphate, you're adding a phosphate. So that's not isomerization. That is a kinase that is doing it. So this is phos, right? So make sure you understand that, okay? All right. So the formation of 3 carbon units also involves substrate level phosphorylation, okay? Substrate level phosphorylation does not need oxygen. So up to this point, all these, this first step, the second step, and the third step of glycolysis. So 3 steps so far have occurred, and we have not produced ATP yet, right? But we have not produced ATP, but we have used 2 ATPs, okay? So glycolysis invests. So meaning, in other words, if you don't have any ATP in your cell, if you don't have any ATP in your cell, you cannot undergo glycolysis, because to have glycolysis, you have to invest. So to make more ATP, you have to invest some ATP. It's like when people say to make money, you have to have money, right? You have to, or to make more money, you have to have some money. So it's the same as invest. So it's the ATP, okay? So ATP has to be present in order for the glycolysis to occur. So in the absence of ATP, you cannot form glycolysis, because glycolysis uses 2 ATPs to form 4 ATPs, but we have a net production of 2 ATPs, right? So you have to understand that. So we have used, 3 steps. The first 3 steps of glycolysis do not produce any ATP, but they use 2 ATPs, okay? Now, the second stage of glycolysis involves the cleavage of the 6 carbon. So now you have formed, so the first 3 steps have formed fructose 1,6-bisphosphate. And then, so the second stage is going to break down the fructose 1,6-bisphosphate to 3 carbon sugars, followed by isomerization, okay? So this is the second stage of glycolysis, the formation of 3 carbon units. And this second stage is the substrate level phosphorylation. Now, the generation of 3 carbon units from 6 carbon sugar is characterized by enzyme aldolase. So if you are going to form 3 carbon molecules from a 6-member ring, you're going to need aldolase. Okay. So in this reaction, so the 2 3 carbon members is dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Please make sure you remember that. Okay, very important products here and I mentioned earlier. All right. So the formation of 3 carbon units, which is also called the substrate level phosphorylation, that's the second stage of glycolysis. Okay, the glyceraldehyde 3-phosphate is generated in this reaction and can continue directly in the glycolytic pathway. The dihydroxyacetone phosphate must be converted to glyceraldehyde phosphate in order to continue in glycolytic pathway. Okay. Now, make sure you understand that. So if you form glyceraldehyde 3-phosphate, this can continue directly into glycolysis. I mean, into other pathways of glycolysis, right? Because this can be broken down, but because also the hydroxyacetone that is formed, this dihydroxyacetone has to be converted to glyceroldehyde. So only glyceroldehyde can continue to form pyruvate, okay? But dihydroxyacetone has to be converted to glyceroldehyde 3-phosphate. And to convert dihydroxyacetone to glyceroldehyde, remember both of them are 3-membered rings. The only difference is the position of the phosphate. So you're going to need isomerization to change the position of the phosphate, okay? All right. So at equilibrium, most of the 3-carbon sugars is in the form of the hydroxyacetone, and this does not form pyruvate, so it has to be converted back to glyceroldehyde. Okay. All right. So very important. So let's look at the structures here. So we mentioned the first three steps of glycolysis form this one, okay? And then the fructose 1,6-bisphosphate is broken down, is cut in half. Aldolase, remember aldolase is going to break down fructose 1,6-bisphosphate. This is very important. Fructose 1,6-bisphosphonate is broken down by aldolase to form dihydroxyacetone and glyceroldehyde 3-phosphate. This glyceroldehyde 3-phosphate here then undergoes isomerization to form this one, because the glyceroldehyde 3-phosphate can be converted, okay, into pyruvate after other steps, okay? So you can convert it to pyruvate. All right. Now, the next step of the glycolysis reaction generates a high potential phosphorylated compound, which is 1,3-bisphoglycerate. So 1,3-bisphoglycerate is a high energy compound. All right. Make sure you know which one is there. Which of the following is a high energy compound? The 1,3-bisphoglycerate. This compound is formed from glyceroldehyde 3-phosphate by the reaction of enzyme glyceroldehyde 3-phosphate dehydrogenase. Okay. Glyceroldehyde 3-phosphate dehydrogenase is the one forming the 1,3-bisphoglycerate. All right. So now, this high energy compound, okay, of 1,3-bisphoglycerate is used to form ATP from ADP. All right. And this reaction is carried out by a phosphoglycerate kinase. Make sure you know which one is forming ATP. And we're going to review that reaction right now. Okay. So, glyceroldehyde 3-phosphate plus NAD plus phosphate. This is the first reaction in glycolysis that forms ATP, okay, or NADH. All right, so you're going to break it down. So glyceroldehyde 3-phosphate plus NAD plus phosphate, categorized by glyceraldehyde 3-phosphate dehydrogenase. Okay, so it's going to form NADH. Okay, so the conversion of glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate forms NADH. Make sure you remember that. Okay, and the 1,3-bisphosphoglycerate is going to be plus ADP. Okay, then you're going to form 3-phosphoglycerate plus ATP. Okay, so the first the ATP that is formed in glycolysis, okay, is by the reaction of 1,3-bisphoglycerate, 1,3-bisphoglycerate into 3-phosphoglycerate. This reaction, when you convert this to this one, forms ATP. This is the first reaction that forms ATP. The first glycolytic pathway that forms NADH is the glyceraldehyde 3-phosphate into 1,3-bisphoglycerate. Very important slide that you're supposed to memorize here. Okay, important stuff here. So pyruvate. Okay, so pyruvate is the last part of glycolysis. Involves the formation of pyruvate and another molecule of ATP. Okay, the first molecule of ATP was formed by conversion of 1,3-bisphoglycerate into 3-phosphoglycerate. Okay, you form ATP. Now the second part of ATP is when you're producing pyruvate. All right. So this formation of the second ATP is, or formation of pyruvate, is accomplished by the rearrangement of the 3-phosphoglycerate to form 2-phosphoglycerate, followed by dehydration to form phosphoenopyruvate. Another important molecule that you're supposed to know, phosphoenopyruvate, right? So, and then finally, the final nearly irreversible reaction is the formation of ATP, substrate level phosphorylation, okay, and pyruvate, which is catalyzed by the enzyme pyruvate kinase. So, formation of pyruvate is catalyzed by pyruvate kinase. Okay, let's look at the reaction. Okay, so here you have 3-phosphoglycerate. Okay, which for you to form 3-phosphoglycerate, you have to form one molecule of ATP already, right? So we already have one molecule of ATP. Now, phosphoglycerate, so we already have one molecule of ATP. Now, phosphoglycerate mutase, okay? Phosphoglycerate mutase is going to shift the position of the phosphate to a different position, and it forms 2-phosphoglycerate. And then the enzyme enolase. Remember, this enzyme is also found in bacteria, right? And that is what the fluoride blocks. Remember? So fluoride blocks the glycolytic pathway by blocking the enzyme enolase. Remember that question, right? This is the enolase, okay? So enolase is going to change the 2-phosphoglycerate, okay, 2-phosphoenolpyruvate. Phosphoenolpyruvate. So to form phosphoenolpyruvate, you need enolase because here is the name. Eno. So you already know that enolase is already involved, right? Phosphoenolpyruvate is formed. Now, look at that. Phosphoenolpyruvate is converted to pyruvate by pyruvate kinase. And this is the second ATP. Now we have 2 ATPs, right? So far in glycolysis. Now, when we say the total amount of ATPs formed in glycolysis is 4, is because this reaction has to go through twice, because remember you have formed 2 molecules, right? You break down 6 to 2 by 3, right? So you still have another one molecule. So if you multiply here by 2, and then you have 4 ATPs, right? Because you break down 1 molecule of ATP to form 2 molecules of pyruvate, okay? All right. So, question. Substrate level phosphorylation occurs when ATP is formed from ADP in the absence of electron transport. That is true. So far, we have not talked about any electron transport, right? So this is true. Make sure you remember that. Substrate level phosphorylation does not involve oxygen. And substrate level phosphorylation occurs in cytoplasm because glycolysis is occurring in cytoplasm, right? So another question. Fructose 1,6-bisphosphate is cleaved, is broken down by the enzyme aldolase to form dihydroxyacetone phosphate and which one? Remember, this is broken down to form 2 molecules. So dihydroxyacetone is one of them, and the other one, which I keep talking so much about, is glycerolaldehyde 3-phosphate. Okay, so our answer is this one. All right. So this is another one. Which of the following steps? Okay. Which of the following steps in glycolysis results in the formation of NADH? So we say that the first 3 steps of glycolysis don't produce anything, right? But the second 3 steps, then they produce NADH. So when you convert the first 3 steps, they produce fructose 1,6-bisphosphate, which is then converted, okay? And the first conversion of fructose 1,6-bisphosphate is going to form NADH. Okay? So, NADH. So, in our answer here, so the first one is going to be the breakdown of fructose 1,6-bisphosphate to glycerolaldehyde 3-phosphate. All right? This one here. All right? So that is our answer. But okay, so that one is broken down to that. So when NADH is formed, okay, when NADH is formed, actually it's formed by this one. So when NADH is formed, the first one actually which forms the NADH is this one. Okay, so the fructose 1,6 bisphosphate is broken down first by the aldolase, okay, to form fructose 1,6 to form glyceraldehyde 3 phosphate and the hydroxy acetone phosphate. But this does not form NADH, but it's the 3,6 bisphosphate to 1,3 bisphosphoglycerate that forms the first ATP. Okay, so understanding this reaction is critical. So our answer is this one. Okay, so you have to make sure you understand that. So the G3P to 1,3 bisphosphate, okay, bisphosphoglycerate is the one which forms NADH. Okay, so key important point here. All right, another one. So this is the end of the review. We're going to have the second uh, glycolysis lecture review. Okay, make sure you look out for that and make sure you review. So review the glycolysis one before you proceed to two. Okay. All right. If you have any questions, please don't forget to email. You can email me here, or you can send us an email or message on Facebook. Okay.